Hey y'all. So we're in week 3, and I'm going to be doing lecture 4, coming at you with immunology and lab techniques for epidemiologists. I think probably a few of you have microbiome backgrounds or lab backgrounds, so you're going to know about some of the stuff already, maybe. But we're going to be going through a little bit of it, just for everybody's sake. So we're going to be talking about some immunology things, a little bit on phylogenetics, some primer on clinical microbiome, and some ID specific laboratory methods at the end. And I'm going to break this up into 4 different pieces so they're easier to chew on. So first, I want to bring you some terminology. For example, immunity. It's something that we sort of commonly have a handle on, but a formal definition would be the ability of an organism to recognize and defend itself against specific pathogens or antigens. Um, when we talk about immunity, we're talking about immunity to something, not just general immunity to something. Antigens are molecules from a pathogen or foreign substance that provoke a specific immune response. So it's basically a foreign object that comes into the body, and the body has ability to recognize it as being from outside and can respond to it in kind. And that's what the immune response is, the production of antibodies and the generation of specialized lymphocytes against specific antigens. So, when a foreign body comes in, it's recognized as being foreign, and the body reacts to it and can produce antibodies to allow it to recognize that particular antigen in the future. So host defense mechanisms. The immune system, quote, is more complex than just simply antibody responses. It also, it's like, you think of the body as a castle, and there's like a moat outside. So what invaders have to cross the moat. There's a wall it has to get through. And then once it gets inside the castle, it has to fight troops or whatever. The immune system of humans and mammalian organisms in general starts with the skin. That's really the first line of defense against invaders. It's the skin's function. It's to protect us, not only to hold us together, but to protect us from outside invaders. Also, we have mucous membranes, in the nasal cavities, in the mouth, and in other areas that also work to protect the body from outside invaders by sticking it to things and then expelling it. And the secretions of skin and mucous membranes, and actually very complex. The skin is not just like sort of a vinyl coating. It actually has excretions, and also, in addition to that, it also has a nice a microbiome that works to provide defenses against invaders as well. The second line of defense of the non-specific defense mechanisms is the phagocytic white blood cells, which go out and basically consume things, antimicrobial proteins, and the inflammatory response. So, the generation of fever, for example, to raise the temperature of the body to fight off certain invaders. Specific defense mechanisms. This is what we formally think of the immune system as the third line of defense, and that's lymphocytes and antibodies themselves, which provide specific defense against specific invaders. So, immune system, I sort of take a broad view of what that is actually more complicated and deeper than just when we think of lymphocytes and antibodies. So types of immunity. There's innate or genetic immunity. And so this is the immunity that you're born with. It's genetically predetermined. It's written into your genes. It may be due to a lack of receptors or other molecules required for infection to occur. So the body, like, you know, evolution, basically, if there's an invader and it successfully can invade and cause disease and kill this particular type of host and not this particular type of host, then the genes that prevent it from either killing the host or allowing access get selected for successive generations. And so that can be when we see the phenomena of there being a lack of receptors or other molecules required for infection to occur. Examples of this would be innate human immunity to K10 distemper. We're immune to it. We just don't have the receptors to allow it to enter and replicate. Immunity of mice to polioviruses. Mice can't get polio. We can, because they lack the receptors to allow that. Acquired immunity is immunity that the host develops over a lifetime, and it's not genetically predetermined. It may be acquired naturally through exposure to pathogens or artificially, for example, through vaccines. Development of immunity to measles in response to infection or vaccination. It could be both, really. It's really an experiential immunity. Antigens, most are proteins or large polysaccharides from foreign sources, microbes, capsules, cell walls, toxins, viral capsids, flagella, etc. And then non-microbes, pollen, egg whites, and red blood cell surface molecules, serum proteins, surface molecules, and transplanted tissue. An epitope is a small subpart of an antigen that interacts with an antibody. And they're in capsules, cell walls, toxins, all these things. And any given antigen may have several epitopes, and each epitope is recognized by a different antibody. Naturally acquired immunity. Again, immunity obtained in the course of daily life. So naturally acquired active immunity are things like antigens or pathogens that enter the body natural. The body generates an immune response to them, and immunity in some cases may be lifelong or just temporary in the case of influenza and intestinal infections. Naturally acquired passive immunity, antibodies pass from mother to fetus via placenta or breastfeeding. And there's no immune response to antigens, and this is usually short-lived. It's generally gone by the time a child is 6 months. But this allows sort of a window of protection to protect children until they can be big enough to resist pathogens on their own. So we see this a lot with malaria, for example. Mothers transfer antibodies to babies, and then for about the first 6 months or so, babies are protected, and then they go off on their own and have to form their own antibodies and immune response to it. So that first 6 months is kind of a necessary window, because otherwise, infants would die soon after birth, because as soon as we exit the birth canal, we're instantly assaulted with millions of different kinds of pathogens. We don't really get a break. Artificially acquired immunity, obtained by receiving a vaccine or immune serum. Antigens are introduced via vaccines through immunization. This is an artificial case, and the body generates an immune response to that. Immune response for some can be lifelong, like the oral polio vaccine, or temporary. As you know, you got to get stuck with your tetanus vaccine every 5 or 6 years. So it just depends on the pathogen, really. Artificially acquired passive immunity, preformed antibodies and serums are introduced into the body by injection. This would be immunoglobulin therapy. Immune is very short-lived, half-life 3 weeks, and the host immune system does not respond to antigens during that period. Antibodies recognize specific antigens. You can kind of think of this as like The body sort of represents, remembers the shape of the invader, picks a part of it. So the next time that comes around, it can recognize that shape, and then raise the alarms when that pathogen is there again. So, I mean, look at this picture. There's an antigen here, and epitope, antigenic determinant, and the binding site is recognizing that. So dual tracks of immune response. There's humoral, antibody-mediated immunity, and then cell-mediated immunity. I will say that you cannot get immunity from a bottle like this. It's very bizarre. Often hear, talk about stimulating the immune system. And I'm always have thoughts of autoimmune disease, where the immune system is overly stimulated, and how we don't want that. So, yeah, these quack medications are just that, Humoral antibody-mediated immunity involves the production of antibodies against foreign antigens. Antibodies produced by the subset of lymphocytes called B cells, and antibodies found in extracellular fluids like blood plasma, lymph, mucus, and etc., and the surface of B cells. These defend against bacteria, bacterial toxins, and viruses that circulate it freely in bodily fluids before they enter cells. So we're going to catch them before they enter cells and start replicating. Cell-mediated immunity involves specialized set of lymphocytes called T cells that recognize foreign antigens on the surface of cells, organisms, or tissues. T cells regulate proliferation and activity of other cells of the immune system, B cells, macrophages, neutrophils, etc. They defend against bacteria and viruses that are inside the cells and inaccessible to antibodies. Once they enter the cell, they're sort of undercover in there. And this would include, this also defends against fungi, protozoa, and helminths, and any other cells, cancer cells, transplanted tissues. Epitopes are antigen regions that interact with antibodies. It's basically like the surface that is recognized by the antibodies. And you can see here they got this little picture with different shapes. And here's, antibody A is trained to recognize this shape. Antibody B is trained to recognize this shape. Antibody C recognize this shape. Unfortunately, I don't have the animation, so we're going to get rid of that. I'll post that online. Main antibody classes. So like if you are suspected of having something like brucella or Q fever, for example, you might get tested. They might test you for what's called IgG and IgM antibodies. So IgG is 80% of serum antibodies total, and they're generally located in the blood, lymph, and intestines. Half-life in serum is quite short, 23 days. Placental transfer, yes. You can transmit IgG through placenta. IgM antibodies are the first antibodies produced during an infection, so they're quite few, and they tend to have a shorter half-life. They disappear quickly. So often when we're testing to see if there's an active infection, we might look for IgM antibodies because that implies an initial reaction to a pathogen. IgG will sort of tell you about infections farther in the past. IgM antibodies are not transferable by placenta. So the dynamics of immune responses, antibody titer, is the amount of antibody in serum. It's measured in serial dilutions. After initial exposure to an antigen, no antibodies found in serum for several days. There's a gradual increase in titer, first of IgM and then of IgG is observed. Most B cells become plasma cells, but some B cells become long-living memory cells, and this gradual decline of antibodies follows. We got a picture coming up. Secondary response, subsequent exposure to the same antigen displays a faster and more intense antibody response. Increased antibody response is due to the existence of memory cells, which rapidly produce plasma cells upon antigen stimulation. So here's a picture of how the body reacts. It's the first exposure to antigen A a little bit after day 0. Antibodies to A increase during the primary immune response. There's a secondary exposure to antigen A, but the first exposure to antigen B, this leads to a second immune response to A, and there's a immune response to B. So your reaction to pathogens is not just to one pathogen. It can be to many. It can be quite complex and last over a long period of time. And I sort of wonder what pathogen this is supposed to be. Kind of don't know. It doesn't say. Human parvovirus B19 infections. Like is this all the same time scale? And here's the symptomatic sequence. Starts fever, then drops off, and then rash and arthralgia comes. And you can see that the antibody responses sort of differ over this time. We get initial IgM increase and drops off because you remember the half-life is quite short, and IgG goes up and remains for a longer period of time. IgG, excuse me. So immunity, the individual and population level. Individual level, it's complete, prevents continued new disease, usually appears rapidly, is protective and sterilizing and long-lived. In the case of complete immunity, not all pathogens lead to complete immunity. Some lead to partial immunity, like Plasmodium for malaria is one that only imparts partial immunity in hosts, and this is a real problem in that people need to be continually exposed to keep immunity to malaria. So when they go and live for several years, like in a non-malarious region, immunity can drop off, so they're again susceptible when they go back to, for example, their visit family and friends in the countryside of Kenya, after living for decades in the States, for example. Again, acquired, passive, active, talked about that. Complex immunology and complex epidemiology. General response to malaria antigens in endemic areas. Really quite complicated here. IgG drops off soon, deaths go up. These are age of children. Risk of death goes up. Parasite rate goes up until about age 10, then drops off. Antibodies go up. And again, this is sort of, assuming that this person until 25 is being repeatedly infected. There's nothing to the idea, that simply being infected, once or twice in infancy, and when they're a toddler, imparts lifelong immunity. That does not happen with malaria. But it just becomes complicated to assess the epidemiology of things like malaria, particularly when you consider that increased immunity to malaria means the symptoms are less, but that doesn't mean they're not being infected, for example. And I think we'll get into this a little bit later. And malaria is interesting, tends to be quite severe in children, leading to high levels of mortality. But, as people grow older and are infected repeatedly, risk of mortality declines to almost nothing. But they're still infected and still able to transmit to mosquitoes and still contributing to community transmission. So we're going to talk a little bit about malaria later. Yeah. So vaccines are important because they have two effects. First, they provide protection to the individual against being sick, and they also provide population protection because that person cannot transmit. Um, so it's similar to what masks are kind of like this. They have two effects: a direct effect and indirect effect. A direct effect to the individual and an indirect effect to the community. The particular effects can depend on the contact behavior of the population, cluster groups of vaccinated persons or cluster groups of unvaccinated persons, protected individually from infection. This, like, vaccines are really, like the best way to protect a population from infection, both because they protect the individual and reduce transmission in the community, and can often extinguish transmission entirely. So we're going to talk about direct and indirect effects a little bit later. So moving back to R naught, again, R naught is the number of cases that arise out of a single case of disease, and it's the measure of potential for an infectious agent to spread in a host population. Abstraction, it's impossible to calculate in practice. I mean, we throw around numbers all the time, but they're estimates and they vary with different places and populations, but they're useful for informing how severe, how serious to take a disease or how not serious to take a disease. Will this disease continue or burn out? I mean, they're estimates. Remember when COVID came out, there were sort of wild estimates of what the R naught of COVID was, and some people were sort of banding them about on Twitter or whatever, and causing panic in people, anxiety in people, which on the one hand it's important information. On the other hand, it's like, well, R naught is an estimate and is to be taken with a grain of salt, and we can't just directly calculate it exactly because it's so contextually dependent and even host dependent, even pathogen genotype dependent in a lot of ways. So I want to talk about that later. I specifically want to talk about COVID-19 and R naught. So I think I'm going to bring that up in a little bit. If you were to calculate R naught, it's just an estimate. It's probability of risk of transmission per contact times the number of potentially infectious contacts per unit time times the duration of infectivity or infected persons per unit time. So, if you have estimates of these numbers, then you can get an estimate of R naught. Greater R naught generally means infection more likely early in life. Therefore, childhood diseases all have high R naught and most confer lifelong immunity. So, I mean, you can think of it this way. It's like measles, because one of the reasons, because measles imparts lifelong immunity, it has to infect as many people as possible as quickly as it can in order to maintain its own survival. Otherwise, it can't keep transmitting in a population because it only gets one chance, basically. So measles has, it's like wild estimates, R naught of 18 or 19. So, up to 18 or 19 people arise from each case. So incredibly infectious. And part of the reason for this is it has, evolutionarily it's pressured to do that because, we do mount a successful lifelong immune response to that particular pathogen. R naught can also equal 1 plus the average lifespan of individual in the population divided by the average age of infection. A lot of you, I don't know how old all you guys are. I'm just assuming that I'm older. I still remember the days, when measles and, mumps, and all these things were running around, and it was sort of just a fact of life back then, that you would get these things. Didn't really question it. I had most of them. These days, younger kids, are sort of they're the less that you see these diseases less because there's been vaccines for decades now, and we sort of successfully suppress transmission within the population for it. So I don't know why I say that. I guess it's just interesting because I just remember it was just sort of it was a given that you were going to get these things, because they were so infectious, and just contact with some kid who had chicken pox meant that you would have it. Immunity itself reduces R naught, and this is one of the reasons R naught is so complicated, is because the host also introduces factors in this. How well we're able to defend ourselves against pathogens, can reduce the number of new cases that arise from each case. Proportion of population immune P reduces new cases as R naught minus R naught times P. I'm not going to test you on any of these formulas. As P increases, new cases decrease, and eventually R naught minus R naught times P becomes less than 1, which means that the minimum P necessary to reduce new cases below 1 becomes P greater than R naught minus 1 divided by R naught. This forms the basis for vaccination thresholds required to reduce transmission in new cases. Again, it's an estimate, and we're just it's just an estimate to inform policy. We see all this talk now about herd immunity. It's an old concept, originally described about 100 years ago. However, I mean, people have their sort of own ideas of what herd immunity is. They all relate to number or proportion of immune individuals within a specific population needed to reduce R naught to 0, to less than 1 to stop community-wide transmission. Now, you often hear about this with COVID-19, talking about herd immunity, but the honest truth is that we don't know what that would be. We don't know enough about immunity to COVID-19 to even really start estimating what a level of herd immunity might be required to stamp out community transmission. So if you see pundits on Twitter or whatever, talking about herd immunity, well, I sort of say that most of them don't know what they're talking about, because we don't even know what that would be. Quantitative cases, proportion immune and resulting reduction of cases due to fewer susceptibles and infectious persons. The total, in qualitative terms, the total resistance of a population, implying some level above which transmission cannot occur. And there's a formula that sort of defines it. But again, we don't know what this would be at this point that I'm doing this lecture. So, like here's a herd immunity required based on different levels of R naught. And obviously, if your R naught is lower, you require a lower number of people to induce community-wide, to break community-wide transmission. If your R naught number becomes very, very high, like 50, I'm kind of wondering what that might even be. Then it becomes very, very difficult to stamp out. So, here's a basic reproductive number for various diseases, pathogens. Flu A is quite low, but measles is very high, and that's why we recommend that all kids, who can, become vaccinated for things like measles, because it's so difficult to control, because R naught is just so high. It's like a larger number of people required to stop community transmission. So R naught is not constant. We can't think of it as constant within or between populations. It varies by season, population structure, age and density, calendars, social clustering, geographic clustering, individual immunity or population groups. You see wide ranges of what R naught could be all around the world, given different contexts. Specific impacts of heterogeneity. Well, everything in epidemiology depends. when you start talking with regular folks out there and they ask questions and you respond with "it depends," they generally don't like that. But it's just the truth. Like everything depends on something. Um, so you can look over this table on your own. Basically, there's different variables and assumptions, and then different implications for herd immunity, including social structure, seasonal changes, age, all these different things. Health program impacts, some implications for herd immunity. You have to vaccinate at times of lowest R naught. You would want to increase population level immunity while the getting's good, while transmission intensity is low. Target high density populations in most cases. Vaccinate following waning of maternal immunity. That's why you're vaccinated in infancy for a lot of things. And these have practical implications for health campaigns. So that's all I wanted to talk about for this lecture. I'm going to move on, I think tomorrow, and put up the second one. Thanks very much. If you have questions, hit me up on Moodle or by email or whatever. Thanks, y'all.